Hello, everyone, and welcome to today's keynote webinar titled Functional Discovery of T-Cell Receptors, TCRs, for Antigen-specific and Unconventional T-Cells using nanovial technology. This webinar is part of the third annual event in the Immuno-Oncology Virtual Event Series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits image located on the right of your screen and follow the process to obtain your credits. All right, now let's get started. I'd like to now present today's speaker, Dr. Dino Di Carlo, Armond and Elena Hairapetian Chair in Engineering and Medicine, Professor and Vice Chair of Bioengineering, Professor of Mechanical Engineering, California Nanosystems Institute, Johnson Comprehensive Cancer Center, University of California, Los Angeles. For a complete biography on our speaker, please find it on the left of your screen. Dr. Di Carlo, thank you for joining us. You may now begin your presentation.
Great. Well, thank you, Kaylee, and thank you, LabRoots, for giving me the opportunity to share the work we're doing. And in particular, the work we're doing on discovering TCRs, so T-cell receptors, which are the affinity elements for detecting diseased cells, cancer, and are being applied for engineered cell therapies. And the big take home for this talk is how we can apply this new nanovial technology and equipment that's already available in your labs to do function first discovery of T-cell receptors. So that means not just based on affinity, but based on the function that is elicited when those T-cells engage antigen or metabolites that are presented by these MHC-like molecules for unconventional T cells. So that's the big take home. The take home is technology. This nanovial technology can allow you to do function first discovery using equipment that's in your lab, and I'm going to share our journey to showing those types of results. So I'd like to start by what motivates us to develop this technology, and I like to refer to the three pillars of medicine. So molecular therapies, gene therapies, and cellular therapies. We're all very familiar with molecular therapies. These are kind of the bread and butter of the pharmaceutical industry for the last decades, small molecule therapies, and more recently, biologics and monoclonal antibodies. And I think these have now kind of taken over as kind of 5 out of 10 blockbuster drugs are now in this biologics category. Gene therapies, we're seeing a lot of excitement in gene therapies and new approvals. I think we had a hemophilia gene therapy approval most recently. And then cellular therapies, which I'm going to focus most on today. And what's really exciting about cellular therapies, unlike molecular therapies, is that cells can respond and interact with their environment. They can sense things. And then based on sensing those things, they can create a response. So we've seen now, I think, 7 approvals of chimeric antigen receptor T-cell therapies, and there's a lot of interest in also using engineered T-cell receptors to target, for example, intracellular antigens, or to target metabolites or other lipids that in these unconventional T-cells can target. And that might be very important for cancer as well as autoimmune diseases. So generally, we're really excited by this nascent field, although really rapidly developing field of cell therapies, and developing tools to help us better find these receptors, like chimeric antigen receptors and T-cell receptors, that are going to be the most functional, especially when we want these cells to be functional against maybe solid tumors or an autoimmune condition. And we focus in particular on developing technologies that allow us to characterize what cells secrete. And so why is that? Why do we think this is important? Well, cells secrete a number of different proteins. There's actually predicted to be greater than 3,000 secreted proteins from the human genome. And that's a similar amount as proteins that are presented on the surface of cells. And they have huge functions. So, huge numbers of different functions. We have cytokines that are basically communication factors to other cells and cause them to change their behaviors. You have antibodies that can act to neutralize, opsonize, or tag through opsonation, tag for killing or other elements. You have proteins that create matrix. You have proteins that are secreted to degrade matrix. You have growth factors, right, that change the behavior and growth of other cells. And you have things like extracellular vesicles, which are emerging area of cell communication factors that have a whole package of different signals that it can deliver to other cells. Unlike proteins that are on the surface of a cell, secretions play an outsized role in how cells interact with their environment. The way I like to explain this is, a cell can whisper to the next cell through its cell surface receptors. It can only, interact with the very near neighboring cells, while it has like a megaphone with secreted factors that can span out to cells that are not just near it, but very far away through transport throughout the body. And so secreted factors are huge, play a huge role. In particular, they play a large role in how T cells achieve their function. So we have, for example, cell killing factors like granzyme B and perforin, but we also have cytokines that are released to really change the environment, draw in other types of immune cells. And so actually the function of these cells is really intimately connected to what they secrete. But we really don't have great tools to analyze and sort cells based on this secretory function. And so that was kind of one of the bottlenecks that we were trying to address with our technologies. And so why is that? So if we look at how we do biology, most biology is done in vessels like petri dishes, well plates, or even 1536 well plates. But even at those volumes of a 1536 well plate, we are thousands of times larger volumes than the volume of an individual cell. And why is that an issue? Well, if an individual cell is releasing things into its environment, that gets diluted out into that large volume of the well and becomes very difficult to measure things accurately when it's diluted out to very low concentrations. So, there's been tools that have been developed to try to reduce that volume, to accumulate those secretions from individual cells to higher concentrations, like microfluidic devices, droplet generators, and microwell arrays, optofluidic approaches. But what I've learned through my time in commercializing microfluidic devices is that when you introduce these new types of devices, they usually come with a new instrument. And that is very particular for that one niche application. And that is a barrier for adoption of those technologies more widely. And also in droplet-based approaches, we don't have a solid phase that would be important if we want to look at secretions, capturing those secretions, and performing, for example, sandwich-type immunoassays, or even using the solid particle phase to interact with T cells in a way like an artificial antigen presenting cell. So this has led to our approach, which we call lab on a particle approach instead of a lab on a chip approach, where the particles themselves become vessels for individual cells. So the particles themselves can isolate individual cells, and the particles can be substrates for reactions and capturing things that those cells secrete. In particular, I'd like to share about one type of particle that we've spent a lot of time developing and working with, which we call a nanovial. A nanovial is a hydrogel particle. It has a cavity that's less than a nanoliter. You can put millions of them into a centrifuge tube, And you can see kind of, these are some part, actually this video isn't going to play, but you can see some of these particles settling down. They have this crescent shape and cross section from the top, but from the top you see kind of a circular shape. So it's basically a bowl-shaped particle. We can make these particles very precisely. Here you're seeing that we have a CVs of less than 1.5%. Here CV is about 2% in the opening diameter. These particles are about 80 microns. We make other particles about 35 microns, which are compatible. I'll share more about this with flow cytometers, as well as 50 microns if we want to look at larger cells or multiple cells. So these nanovials, as I mentioned, they're hydrogels, they're transparent, which makes them very good optically clear particles that we can then also capture things on and measure fluorescence of. So we can functionalize the surface of these hydrogels to both capture cells specifically, as well as we can add antibodies, for example, to capture secretions that are released from those cells. And then we can label those secretions. And the particle is also compatible. It can be analyzed at very high throughput with traditional instrumentation like flow cytometers, like single cell sequencing instruments. And this is a key aspect of technology. As I mentioned at the beginning, we're motivated to not have another new instrument that would be a barrier for entry, a barrier to use these types of sophisticated technologies widely by the community. So we wanted something that could be usable with instruments that folks already have, for example, flow cytometers, FACS systems that are already available in labs. So how do we use these types of particles? This is kind of a general workflow for how do you load cells and analyze secretions from nanovials, cells on nanovials. Basically, we mix cells and nanovials in a tube, mix them up, allow the cells to bind, could be specifically to some antibody on the cell, or it could be, in the case of these T cell workflows I'm going to talk about, actually a peptide MHC molecule that's on the nanovial, or a MHC-like molecule presenting a metabolite. We can then also include antibodies on the surface of the nanovial to capture the secretion. So when the cells and nanovials are incubated, the secretions are released. They're kept within this cavity because you prevent convective flows. So most of the transport out of the cavity is minimized. And so you get accumulation of secretions inside that cavity and captured on the surface of the nanovial. Those secretions can then be stained, for example, with fluorescent antibodies or oligo-tagged antibodies to form a sandwich. And then they can be analyzed by flow. So high secretors can be sorted, let's say, out. You can then regrow those cells downstream, characterize productivity or other cell killing or other aspects. Or you can go directly into a single cell sequencing workflow where you're characterizing, for example, the T cell receptors. And I'll talk about that workflow, or transcriptomes, or both. As well as, because we have this oligo-tagged antibody, we can also characterize the secreted proteins in a technique we call seq-seq, or secretion-encoded single-cell sequencing. And so we can link the secretions, the amount of secretions, with the TCR sequence, for example. So that's really exciting because we can start to rank TCR sequences based on the amount of secretions, amount of cytokines that are released by a cell that is activated, that has that particular TCR. So I'll dig into that more later on in the talk. So we refer to this lab on a particle approach as kind of a microfluidics 2.0 approach, because we use microfluidics to make these particles, but that's done once. And the kind of the end user of the particle, whether it's our collaborators or others, they have to have no microfluidics expertise. They don't need a microfluidic instrument. They don't need a syringe pump. They need nothing, just the particles themselves and the instruments to analyze them, like flow cytometers. So this is a relatively nascent field for us. We have started publishing in this field maybe 2 years ago in 2022, and really had some more publications come out in the last year and even earlier this year. And this is actually a lot of the work that I'm going to talk about today is also found in this paper in Proceedings of the National Academy of Sciences. And so really exciting that we're really digging into this field and building it out, and lots of new publications coming out every month or to a few months. Okay, so I want to first talk about our work using nanovials for function-first screening of T cells. And so this is work that was led by my student, Felice, and continued by Savannah and Citra in the lab. And this has been a great collaboration with Mao from Owen Whitty's lab. And also we've started to work with Lily Yang's lab on the unconventional T cells. So what are we trying to do here? Well, we want to basically find T cell receptors, right? Because these can be engineered into new T cells to create kind of armies to target tumor cells and expand that ex vivo or in vivo. And so the big challenge here that we're trying to address is finding rare T cell receptors that are targeting very specific antigenic peptides that are associated with types of tumor cells. And so, for example, we'll talk about work that we're looking at prostate cancer specific epitopes that are presented by MHC and finding rare TCRs that can recognize those epitopes. And so, the challenge here is this is a needle in the haystack problem, right? There's huge diversity in any particular patient and their TCRs. And, just looking at binding, for example, using tetramers or something to fish out cells that bind is not sufficient. So when you do approaches like that, you get lots of false positives, especially for these very rare TCRs, because tetramers can stick to many different things. And it doesn't tell you about the function of the cell. If it binds, it doesn't say that cell is actually going to be activated when it binds to that particular peptide MHC. So this is where we wanted to really create this function-first approach where we're measuring both affinity and function of a T cell, and then using those two constraints to be really confident that we have T cells that would have TCRs that will be functional. So as I mentioned, there's a number of different approaches to screen for antigen-specific T cells to find TCRs. In particular, peptide MHC tetramer approach. As I mentioned, some of these approaches, though, even though you're using a tetramer, you still have, you may miss low affinity TCRs, which still could be valuable to have. The specificity is not as great because you can get binding off target. But you have the advantage of finding viable cells. You can start to explore getting more functional readouts by doing, for example, intracellular cytokine staining. Although the accumulated cytokines is not exactly the same as what's being secreted, and so it is a kind of a surrogate that can be used. But the challenge here is that when you do this process, you are permeabilizing the cell in order to stain the intracellular cytokines, and that permeabilization process means the cells die, as well as the RNA, it becomes much more difficult to recover. And so recovering TCR sequences, for example, using a single cell connect workflow becomes more challenging. So, and then finally, you do things like surface staining with activation markers, which tells you something about the function, but specificity also can be low in this case. Okay, so what our approach was to truly try to combine the best of both worlds, where we want to do an affinity-based selection of T cells on our nanovials, followed by a functional screen of cells that are activated by peptide MHC on the nanovial and are elicited to secrete cytokines, or interferon gamma in this case. We also look at granzyme B, TNF alpha, IL-2. It's a general approach. So how does this all work? So first thing we need to do is functionalize the nanovial to make it like an artificial antigen presenting cell. So we put peptide MHC with a particular peptide MHC monomer that's biotinylated on a streptavidin-coated nanovial. this creates huge amounts of peptide MHC monomers compared to a tetramer, right? So you have huge avidity effects. We also put the capture antibody for our secretion, in this case, interferon gamma. And then finally, we put an oligo barcode. So we're putting a streptavidin with an oligonucleotide that tells us which peptide MHC was on this particular nanovial, so that we can do a pool of many different peptides and screen all of that in parallel. Finally, once we have these, once we have this pooled set of nanovials, we can then load T cells. In a typical experiment, we're looking at like 20 million T cells loaded onto nanovials. Some very small fraction of those are antigen specific that will bind. We can capture the cells specifically based on their TCR interactions with peptide MHC. We then wait 3 hours for those cells to secrete. So they are stimulated through this interaction with the nanovial to secrete cytokines. So interferon gamma, granzyme B, or TNF alpha, or other effector molecules like granzyme B. After 3 hours, we then wash and add a detector antibody as well as antibodies to cell surface markers on the T cell. So CD3, CD8, we're looking at cytotoxic T cells that are also secreting interferon gamma and that have also bound specifically to the nanovial. We can then sort out this subpopulation of high secretors and then go downstream, look at, we can look at proliferation of those cells, but we also, and most of the workflows I'll talk about, go directly to a single cell sequencing workflow where these sorted out nanovials go right into a droplet generator of like a 10x chromium workflow to get single cell matched alpha and beta chains of the TCR. So does this workflow work? So let me just kind of go through some of the initial data to characterize this workflow. So we started off with a engineered T cell that had this 1G4 TCR that's specific to NY-ESO1 peptide MHC. This particular construct also had an NGFR region that we had an antibody to, so we could stain whether that particular TCR was present. And so what we did first is like, okay, do the cells bind specifically? So we do see selective binding. When you have peptide MHC concentration increase, we see more binding of the 1G4, but we don't see binding of the untransduced PBMCs. And the nanovials with cells, the ones that are binding, are mostly NGFR positive, right? And then second, we get selective secretion. So cells that are bound, these 1G4 cells that are bound to peptide MHC on the nanovial, they are secreting interferon gamma, while 1G4 cells that are bound just to CD45 antibodies on the nanovial, they don't secrete interferon gamma. So you're not seeing that interferon gamma signal. This is after 3 hours of incubation. Okay, so we went, a first kind of experiment just to characterize that this platform was working was to look at some viral peptide MHC molecules, or some viral peptides presented by MHC, and then fishing out some TCRs that are specific to CMV and in EBV. So we had 3 particular subsets of nanovials, 2 peptides for CMV and 1 peptide for EBV. We had barcodes for each of these, and we compared this to tetramer-based approach as well as just an activation marker-based staining approach. And so this is kind of the gating strategy. We look for single T cells that are on nanovials, so based on a viability dye, and then the CD3, CD8 positive cells, and then the interferon gamma positive cells. And those are all sorted, and then we go downstream to the sequencing. So we put directly these nanovials into the chromium, the 10x chromium system to form the emulsions and create the cDNA libraries. And in this case, we compared, as I mentioned, tetramer, the multiplex nanovial, CMV1, CMV2, EBV, and then CD137. So tetramer-based approach, we had less hits, about 800 sorted events, about 6,000 sorted events for nanovial and CD137. number of cells. When we put in 800, we get about 287 out. Nanovials, you put in 6,000, we get 1,500 out. So if you look at the end product of then these productive VJ spanning pairs, we get about 1,400 for nanovials, 90% or so, which is similar or better than the other approaches. So the nanovials themselves don't interfere with the single cell sequencing workflows, single cell library generation, and then downstream sequencing. Okay, so what came out of this? What kinds of TCRs? Well, we could compare across the 3 different techniques, and what we found is that the tetramer approach had TCRs that came out, but only a few, and actually the nanovial and CD137 approach had more, and that they overlapped, mostly overlapped, so 15 overlapping ones, and they also subsumed the 2 from tetramer-based approach. And we can do a little bit more. We can then re-express these TCRs that were recovered, and what we find, and this is an NFAT GFP JURCAT reporter line and that are exposed then to antigen presenting cells. What you see is that the tetramer-based ones we discover they are GFP positive generally. However, we find 6 additional nanovial-based CMV1 specific TCRs that are also showing positive GFP signal, as well as EBV specific ones that were encoded with the other peptide. We can then also compare this between CD137 workflows and nanovial-based workflows, again with the NFAT GFP reporter. We can see generally we get positive GFP signal, some nanovial specific TCRs that weren't picked up with CD137 and vice versa. And then we also went repeated this, but also with PBMCs and looked at interferon gamma secretion. And again, we are able to find a number of different TCRs using the nanovial-based approaches that overlapped with the CD137-based approach and some unique ones. But the nanovial-based approach, unlike the CD137-based approach, we could also annotate which specific peptide those TCRs were recognizing, and we did that with 100% accuracy. So there are no mistakes in identifying that this particular TCR5 was against CMV1, which would require follow-up experiments for the CD137 workflow. Okay, so this was kind of a first test of this workflow, and we're really excited to see that the nanovials allowed us to find, accurately find TCRs. And so we wanted to go on and apply this to much more rare TCRs that are cancer specific. In this case, prostate cancer specific. These are splice variants of particular antigens in prostate cancer. And in this workflow, we then also added the capability to look at granzyme B instead of interferon gamma. So here we're looking at previously identified by our collaborators, PAP22 TCR, and we can see that you elicit high levels of granzyme B secretion on the nanovials when these T cells interact with their cognate peptide MHC. And so that was kind of the first, just, okay, does this work? And then we went and actually looked at patient samples and were trying to identify new TCRs that were going to be specific to prostate cancer peptides. So here CD3, CD8 gate, and then we do our granzyme B gate. And so we did this sort. We then find that we can, so one thing that we did different with this experiment was we also tagged the secreted granzyme B with an oligobar-coded antibody. So we had an anti-granzyme B that had, actually in this case it was anti-APC. So we had labeled the granzyme B with APC. We had an anti-APC with an oligobar code. And so what we could do is after we sorted, you can see there's huge variation in the granzyme B secretion, still about an order of magnitude in this gate. And so after we sorted, we could also annotate the particular TCRs based on the amount of granzyme B level. So The granzyme B negative gate, you can see, has low granzyme B oligobarcode reads. The granzyme B positive gate has higher granzyme B positive reads, and so you can also find some of these TCRs that have much higher reads, a much higher secretion. So this was like the first time that we could link the TCR sequence in this discovery process with the amount of granzyme B and use that to help rank the TCRs. And so that was actually very helpful. What we found is that we found 3 new TCRs, but what we found is that the granzyme B high population, that we had a much better functional recovery rate of TCRs than looking at granzyme B medium or granzyme B low. So this was really exciting to our collaborators, because in previous work, when they're using these other approaches to find TCRs, they may go through 20 or so patients, and maybe 2 to 3% of the TCRs that they actually sequence when they re-express them are actually functional. So if they can get to this 10 to even 30% or more of the TCRs being discovered are actually functional upon re-expression, that's a huge boost to them. So this was exciting, and I think gives you a sense of how this function first discovery can improve the workflows so that you have less kind of garbage TCRs that are being re-expressed and through expensive and long downstream processes, and really can focus on the best TCRs. Okay, just to kind of give you a flavor of where this can go further, we've also incorporated in the nanovial workflow the ability to look at multiple different cytokines. So here we're seeing staining for interferon gamma production as well as TNF alpha production. And you can see some cells that are just interferon gamma producers, some that produce interferon gamma and TNF alpha, and other cells that just produce TNF alpha, and then a large fraction that don't produce any cytokines upon stimulation on the nanovial. So this is exciting. You can start to try to understand, are there differences in TCRs that elicit particular types of functional secretion profiles, or is that not related to the TCR? Is that related to other epigenetic states of the cell? And then we've started to look at beyond kind of these traditional T cells that are targeting peptide presented by MHC. We started to look at unconventional T cells because there's some excitement here that they can recognize non-peptide antigens that could be unique for example, metabolism of tumor cells. They are not HLA restricted. So this means that if you make a therapeutic based off of these, that they can be applied to a larger cross-section of the population. You have a kind of a variety of these types of unconventional T cells that target different types of things like lipids and metabolites. So we're in particular with Lily Ying's lab, we're looking at mate cells, mucosal associated invariant T cells that target MR1, as well as innate natural killer T cells targeting CD1D. So we applied the nanovial workflow for MR1 and CD1D loaded nanovials. Again, we barcoded each of those so we could differentiate. We had interferon gamma as our capture antibody for secretions, and we basically did the same workflow: accumulate load cells, accumulate secretion for 3 hours, sort, and perform single cell sequencing. What you see in kind of just validation of the platform is that when you have iNKT cells that are on CD45, some fraction that get loaded, control T cells do as well, but they don't secrete interferon gamma. Here, when you have iNKT cells on CD1D loaded nanovials, they bind as well. Control T cells don't bind, and you get high levels of interferon gamma secretion. And then we did the same with MR1. Mate cells will bind specifically, and you get high levels of interferon gamma secretion from those mate cells. You can see beautiful little picture of these nanovials, the cells inside, and this blue ring is the interferon gamma in this brilliant violet 421 stain. Okay, so we did this workflow, and as I mentioned before, we also used an oligo-tagged ABC to rank based on secretion. I'm going to briefly just go through some of the results. We're still really in the process of analyzing. This is hot off the press, but we start to find clonotypes that are in TCRs that are specific to iNKT cells, as well as mate cells in these runs. And we're trying to now understand, is there something special about which sequences and which BDJ sequences and genes are being used that relate to the function? So is there anything, cells that secrete more interferon gamma, are there particular subsets of these VDJ genes that are used, or CDR3 sequences? So this is a work in progress, but we're able to identify iNKT-like cells, mate cells, and we're kind of further expanding this, re-expressing and analyzing these cells further. Okay. So to kind of summarize, these nanovial-based approaches, we think they can really help scale the ability to look at secretions and function of individual cells thousands of fold compared to what's out there, the multi-well plate approaches, right? So multi-well plate approaches, you have these large volumes. Nanovials, we have 1,000 times smaller volume, nanoliters compared to microliters. We can make thousands of times more compartments and load cells into each of those. Not each of those. We actually load through Poisson loading, so because we want single cells. So, but thousands of compartments, thousands of times more compartments, and some fraction of those will have cells. And then you can input these into other single cell instruments. So these nanovials can go into flow cytometers. They can go into single cell sequencing instruments. And that enables other functional readouts that you don't have with a well plate. Okay, so with that, I'm happy to acknowledge the team that has been working on this. As I mentioned, Felice, Savannah, Citra, Shreya has been working on our seq approaches. Joe was a Ph.D. student in the lab who started the nanovial work and is now the CEO of a company called Partillion Bioscience, which provides, is producing nanovials and providing that to the community. Rob Di Matteo was also a student, worked initially on the T-cell work, and So Young developed some of the formulations for nanovials. And then our collaborators, Owen Whitty's lab here at UCLA, Mao from Owen Whitty's lab, Lily Ying's lab, and Charlie and John from Lily's lab. So with that, I'm happy to take some questions, and thank you again.
All right. Well, thank you so much, Dr. Di Carlo, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. All right, so let's get started. Our first question here asks, why do many cells that bind to nanovials not secrete cytokines?
Yeah, that's a great question. I think there's two aspects to that. One is the biology, and then one is this aspect that I talked about, where we're using the functional piece of nanovials, the ability to characterize their secretion, as another constraint on selecting the cells that are of interest. So the first point, when we use something very kind of blanket activation through like PMA inomycin, what we see is not all of our T cells will secrete cytokines, and it depends on the cytokine. Granzyme B, we get a large fraction, a much higher fraction of T cells will secrete granzyme B. Interferon gamma is less. TNF alpha is a little bit less. IL-2 is also less, and it depends on the whether they're CD4 or CD8 cells as well. So I think part of it is biology. So it's not all the cells are, may have accumulated those particular proteins or have gene expression programs set up to produce those. And then the second is that you have cells that bind potentially non-specifically. I think it's a small fraction, but you have some cells that will bind non-specifically to nanovials. And those cells, they shouldn't be secreting, right? They're not being activated in a functional way. And so that function-based screen allows us to eliminate those non-secreting kind of crosstalk cells that are binding maybe non-specifically.
Great, thank you. Another question for you here. What is the shortest time you have tested for IFNG and granzyme secretion?
Yeah, so we've done, I think, from like 1 hour to 6 hours and overnight. What we found is 3 hours is kind of the sweet spot. You do have secretion already at 1 hour. So these are probably stores of granzyme or interferon gamma that are already present in the cell that are being released upon engagement with huge amounts of peptide MHC that are clustering TCRs on the nanovial.
Great, thank you. All right, next one here is a two-part question. How many analytes have you looked at per vial, and what do you think the limit is?
Yeah, so I had shown some data on 2 different cytokines, and the limitation really is on the number of colors of a flow cytometer. I think that's the fundamental limitation. The surface area of a nanovial is larger than that of a cell. So you can imagine if you can look at many different cell surface markers using like a 40 color panel, you could look at many different cytokines. The caveat there is that as you start to do that, if you put more and more different types of capture antibodies, then you have challenges, right, where you start to limit the dynamic range of each of those. So you want to kind of have that balance of, okay, which cytokines do I really want to look at and focus on those? Because as you add more and more different capture antibodies, you're going to limit the dynamic range for each of the particular cytokines of interest.
Great, thank you. All right, now how many T cells can you analyze with nanovials, and what rarity of cells can you isolate?
Yeah, that's a great question. So as I mentioned, we are generally running the workflow with like 20 million PBMCs to start with. Of course, not all of those are T cells, and an even smaller fraction are going to be antigen-specific. In the case of the prostate cancer, rare prostate cancer specific T cells and TCRs that we recovered, I think we recovered, I think it was 100, maybe 150 or 200 cells from the sort. And then maybe like 50 of those went through the whole TCR sequencing workflow. And so, you can look at, these are really rare populations. We've also done some spiking experiments where we spiked in antigen-specific T cells into a background of other T cells. And we went down to like 1 in 30,000 spiking, and we were able to recover those spiked cells. That's kind of the level of rarity that we can start to recover.
Great, thank you. All right, moving on here. Have the nanovials already been used for in vivo applications?
So nanovials they're large, right? So they're 35 microns. They're not something you would inject into somebody. They're not approved for any of that. They are in vitro tools to discover TCRs in this case, but also to characterize cell populations. So we've also applied this, for example, to other types of chimeric antigen receptor T cells, CAR T cells. We've applied this to screen plasma cells that are secreting antibodies. But all of those are in vitro experiments, right, where we're using flow cytometers and other tools in kind of the lab setting.
Great, thank you. All right, it looks like we have time for one more question here, so we'll wrap up after this one. Would this discovery workflow be applicable to CAR T cell libraries as well?
Yes, yeah. So instead of putting the peptide MHC monomers onto the nanovial, what you can do is put antigen directly on the nanovials. So we've done this for, like, example, HER2. We've put CD19 on nanovials, and then you can selectively enrich CAR T cells that bind to those antigen And they will also become activated to secrete cytokines. And so that way you can sort out specific populations of CAR T cells that are the most activated. You can also screen libraries of CAR T cells. So what we've done is with a collaborator who had a CAR library with different intracellular domains and wanted to know which was going to be the best to elicit particular cytokines, we could screen out that library and then identify particular sequences and particular intracellular domains that were enriched for, let's say, causing the cells to secrete more interferon gamma.
Great. Thank you so much. All right. Well, that will conclude our session for
I
today.
think there's
So,
another question that came up. Sorry. I saw.
Oh, so sorry about that. Let me pull that up here. Okay. Yep. We can ask this one. This question asks, which is better or more sensitive for TCR discovery, IFNG or granzyme?
Yeah, that's a great question. I think we haven't explored that in a, I would say, rigorous manner. We haven't kind of done side by side. But but what we're finding is that granzyme B is secreted by a larger fraction of cells. Interferon gamma is secreted by a smaller fraction. And so we've been using, for our latest workflows, we've been using granzyme B as the secretion of interest instead of interferon gamma for doing the kind of really rare TCR discovery.
Great. Well, thank you so much. That will conclude our session for today. Do you have any final comments for our audience?
Oh, thank you so much. I appreciate your time and hope this was helpful for you in your research.
Great. Well, thank you again, Dr. Dino Di Carlo, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 30th, 2025, and Labyrinth will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.